Nutrition. Food is any substance consumed to provide nutritional support for the body. It is usually of plant or animal origin and contains essential nutrients such as fats, proteins, vitamins, or minerals. The substance is ingested by an organism and assimilated by an organism's cells to provide energy, maintain life, or stimulate growth. Nutrition may be defined as a science of food and its relationship to health. It is concerned primarily with the part played by nutrients in body growth, development, and maintenance. The science or study that deals with food and nourishment, especially in humans. The word nutrient or word food factor is used for specific dietary constituents such as proteins, vitamins, and minerals. The process of taking in food and using it for growth, metabolism, and repair. Nutrition is the intake of food considered in relation to the body's dietary needs. Good nutrition, an adequate, well balanced diet combined with regular physical activity is a cornerstone of good health. Poor nutrition can lead to reduced immunity, increased susceptibility to diseases, impaired physical and mental development, and reduced productivity, according to WHO, World Health Organization. Dietetics is the practical application of the principles of nutrition. It includes the planning of 4 meals for the well and the sick. Good nutrition means maintaining a nutritional status that enable us to grow well and enjoy good health. Recommended dietary allowances or RDA is defined as the nutrients present in diet which satisfies the daily requirements of nearly all individuals in a population. RDA or recommended dietary allowances is defined as the intake of nutrient derived from diet which keeps nearly all people in good health. RDA takes into account individual variation in nutrient needs and also the availability of nutrients which may vary from diet to diet. This implies additional safety factor amount to estimated requirement to cover the variation among individuals, losses during cooking, and lack of precision inherent in the estimated requirement. Recommended dietary allowances requirements plus safety factor. Adequate intake is a nutrient recommendation based on observed or experimentally determined approximation of nutrient intake by a group of healthy people when sufficient scientific evidence is available to calculate an RDA. A diet that consists of right amount of essential nutrients such as carbohydrates, proteins, fats, vitamins, minerals, okay, and what are required by the body is called a balanced diet. Balanced diet ensures the supply of the entire required nutrient in such the right amount to the body. The basal diet of a person differs from another as the body functions and the overall energy requirement for the body differ from person to person. A balanced diet is essential and vital for the optimal growth and development of the body. Balanced diet helps to prevent deficiency diseases such as protein, energy, malnutrition, vitamin deficiency diseases such as beriberi, scurvy, pellagra, rickets, mineral deficiency diseases such as goiter, anemia. Balanced diet also helps in preventing obesity as it provides just the required amount of energy required. An unbalanced diet may hamper the functioning of the body and also overall health and development may get affected. A balanced diet should be low in cholesterol, sugar, salt, alcohol, and saturated and trans fat. Balanced diet is also known as healthy diet as it includes fresh fruits and vegetables. A balanced diet would be one composed of major food groups carbohydrates, proteins, vitamins, minerals, fats, and water. So, dietitian, a person with legally recognized qualification in nutrition and or dietist who applies the science of nutrition to the feeding and education of groups of people and individuals in health and condition. Balanced diet is a one which provides all the nutrients in required amounts and proper proportions. Food pyramid is a tool to plan balanced diet. Calorie, a unit of heat used to indicate the amount of energy that foods will produce in the human body. The amount of heat required at a pressure of 1 atmosphere to raise the temperature of 1 gram of water 1 degree Celsius, that is equal to about 4.19 joules, abbreviation CAL, also called gram calorie. Here we could see a picture which represent the energy calorie value requirement for male, female, pregnant ladies, lactation time, lactation, infant with various age difference, children, aged persons, like this. Energy requirement: protein, fat, retinol, carotene, thiamine, riboflavin, nicotinic acid, pyridoxine, ascorbic acid, folic acid, vitamin B12, calcium, iron, and their approximate body weights are also shown. Next, digestive system. Living organisms requires nutrients and oxygen for survival and growth, yet we get all these things from the food, but the body cannot utilize the food material in the form as we eat. It has to be broken down into small particles by a process of digestion. Only then it can be absorbed. Then parts of the digestive system. Digestive tract is more than 10 meters long from one end to the another end. It is continuous, starting from the mouth, passing through the pharynx, esophagus, stomach, small intestine, large intestine, rectum, and finally into anus. Accessory organs of digestion: the teeth, tongue, salivary glands, liver, gallbladder, and pancreas. General organization of the digestive system. In general, the wall of the digestive system from the posterior pharynx to the anus has following layers from inward to outside: mucosa, submucosa, muscularis, and serosa. MSMS. But we should know what are this: mucosa, submucosa, muscularis, and serosa. Inward to outside: mucosa, submucosa, submucosa, muscularis, and serosa. Mucosa, submucosa, muscularis, and serosa. From inside to outside: mucosa, submucosa, muscularis, and serosa. There are smooth muscle fibers in the submucosa and two layers of smooth muscles in the muscularis. Outer longitudinal and intercircular layer. The wall of the lumen is lined by mucosa throughout except in esophagus and distal rectum. The serosa continue. Once again, I will be reading this. There are smooth muscle fibers in the submucosa, where is submucosa situated after mucosa and before muscularis region. So, the smooth muscle fibers in the submucosa and two layers of smooth muscles in the muscularis. Outer longitudinal and inner circular layer is there. The wall of the lumen is lined by mucosa throughout except in esophagus and distal rectum. The serosa continues on to the mesentery, which contains the nerves, lymphatics, and blood vessels supplying the tract. Here we could see a picture which represents GI tract wall layers. Serosa, longitudinal muscle, hyenteric plexus, circular muscle, submucosa, submucosal plexus, muscularis mucosae, then mucosa, mesentery regions. Okay. Next, innervations of gastrointestinal system. There are two major networks of nerve fibers that innervate the GI tract: intrinsic and extrinsic, together to form the enteric nerve system. Intrinsic innervations. The two major networks of nerve fibers intrinsic to the GI tract are the myenteric plexus or Auerbach's plexus, myenteric plexus, between the outer longitudinal and middle circular muscle layers, and submucous plexus. Myenteric and submucous plexus. Never forget this too. Myenteric plexus and submucous plexus, or Meissner's plexus in short. We might have learned about Meissner's corpuscle when we talk about mechanoreceptors in body. So just remember that Meissner's word might be the same scientist discovered this, I don't know. So Meissner's corpus plexus and myenteric plexus between the middle circular layer and the mucosa. The myenteric plexus is concerned primarily with motor activity, and the Meissner plexus innervates the glandular epithelium, intestinal endocrine cells, and submucosal blood vessels are primarily involved in the control of intestinal secretions. The myenteric plexus is concerned primarily with motor activity, and the Meissner's plexus innervates the glandular epithelium, intestinal endocrine cells, and submucosal blood vessels are primarily involved in the control of intestinal secretions. And extrinsic innervations. The intestine receives the innervations from the autonomic nervous system with parasympathetic cholinergic fibers, generally increasing the activity of the intestinal smooth muscles, and sympathetic fibers generally decrease the intestinal activity and cause the contraction of the sphincters. Activity of the gut is essentially a parasympathetic phenomenon. When we talk about the movement of gut, we know that peristalsis is a primary process. Before that, extrinsic innervations. The intestine receives the innervations from the autonomic nervous system with parasympathetic cholinergic fibers, generally increasing the activity of the intestinal smooth muscles, and parasympathetic fibers generally decrease the intestinal activity and cause the contraction of the sphincters. Activity of the gut is essentially a parasympathetic phenomenon. Then movement of GI tract. Peristalsis, a reflect response initiated in the gut wall due to stretching of walls by the contents of the lumen. It occurs in all parts of the GI tract from esophagus to rectum. Stretch causes circular contraction behind this stimulus. It occurs in all parts of the GI tract. A reflect response initiated in the gut wall due to stretching of walls by the contents in the lumen. It occurs in all parts of the GI tract from esophagus to rectum. Stretch causes circular contraction behind the stimulus and an area of relaxation in front of it. This wave of contraction moves the luminal contents from oral to caudal direction, not in reverse. Okay. So peristalsis is due to integrated activity of the enteric nervous system. It can be increased or decreased by the autonomic input. Stretch causes release of serotonin, which activates sensory neurons, which stimulate my enteric plexus. Cholinergic neurons cause. So once again, I will be reading this peristalsis. A reflect response initiated in the gut wall due to stretching of walls by the contents of the lumen. It occurs in all parts of the GI tract from esophagus to rectum. Stretch causes circular contraction behind the stimulus and an area of relaxation in front of it. Contraction and relaxation. Okay, this wave of contraction and relaxation waves the luminal contents from oral to caudal direction. That will move that food items probably. So peristalsis is due to integrated activity of the enteric nervous system. It can be increased or decreased by the autonomic inputs, and stretch causes release of serotonin, which activates sensory neurons that activates my enteric plexus. Cholinergic neurons cause release of substance P, and acetylcholine cause smooth muscle contraction. Stretch causes release of serotonin, which activates sensory neurons that activate my enteric plexus. Cholinergic neurons cause release of substance P, and acetylcholine cause smooth muscle contraction. Substance P and acetylcholine cause smooth muscle contraction. At the same time, cholinergic neurons passing in the anterograde direction activate the neurons which secrete NO, VIP, and ATP, causing relaxation ahead of the contraction. Basic electrical activity, or BER. Except in the esophagus and proximal part of the stomach, the smooth muscles of the GI tract has spontaneous rhythmic fluctuations in membranes potential between minus 65 MV and minus 45 MV. This basic electrical rhythm is initiated by the intestinal cells of Kajal, or I don't know how to pronounce this, CAJL. Then stellate mesenchymal pacemaker cells with smooth muscle-like features that send long multiple branched processes into the intestinal smooth muscle. Migrating motor complex. During fasting periods, fasting between periods of digestion, the pattern of electrical and motor activity in the GI smooth muscle become modified so that cycles of motor activity migrate from the stomach to the distant ileum. Migrating motor complex starts with a quiescent phase 1 during fasting between periods of digestion and pattern of electrical and motor activity in the GI tract. Smooth muscle become modified so that cycles of motor activity migrate from the stomach to the distant ileum. Migrating motor complex starts with the quiescent phase 1, continues with the phase of irregular electrical and mechanical activity of phase 2, and ends with the burst of regular activity. That is the phase 3 part. So the MMCs migrate at a rate of about 5 centimeter per minute and occur at an interval of 90 minutes. They clear the stomach and small intestine luminal content and prepare for the next meal. They are immediately stopped by ingestion of food and return of peristalsis and other forms of BER and spike potential. Functions of GI tract. GI tract is responsible for breaking down the food particles and supplying the body with water, nutrients, and electrolytes needed to sustain life, which is brought by the following process: ingestion, including placing food into the mouth, chewing the food, and breaking into smaller pieces by mastication, moistening the food with salivary secretion. Then digestion, during which the food particle is broken down into smaller particles by the grinding action of the teeth and degraded by the digestive enzymes into absorbable nutrients. Then starch, degraded by salivary and pancreatic amylase into disaccharide maltose, trisaccharide maltotriose, and polymers of glucose. It's dextrin. By the action of oligosaccharides in the small intestine, starch derivatives are converted into maltose, sucrose, and glucose. Then proteins degraded into oligosaccharides in the small intestine, starch derivatives are converted into maltose, sucrose, and glucose. Then proteins degraded into proteases and dipeptides by the action of pancreatic enzymes. Fats degraded by lipases and esterases to monoglycerides and free fatty acids. Next, absorption process will be taking place. During this process, the nutrients, water, and electrolytes are transported from the GI tract into the circulation. The digestion process will be taking place. During this process, the undigested food, along with the various secretions and slaughtered off epithelial cells from the GI tract, passes into the rectum and from the feces, which are voided through the anus at periodic intervals. Next, processes of digestion and absorption. Mouth and salivary glands. In the mouth, food is mixed with saliva, and by the process of mastication, large food particles are broken down into smaller ones. Chewing action of the teeth is by the movement of lower jaw against upper jaw, which is brought about by muscles of mastication. The muscles of mastication include masseter temporalis, internal and external pterygoid. Pterygoid. I'm sorry for mispronouncing. Masseter temporalis, internal and external pterygoid, and basinate. I'm not sure about the pronunciation, just read this. This food particle forms bolus and is propelled into esophagus. Processes of digestion and absorption. Mouth and salivary glands. In the mouth, food is mixed with saliva, and by the process of mastication, large food particles are broken down into smaller ones. Chewing action of the teeth is by the movement of lower jaw against upper jaw, which is brought about by muscle of mastication. The muscles of mastication include masseter temporalis, internal and external pterygoid, and basinate. The food particle forms bolus and is propelled into esophagus. 3 pairs of salivary glands are present in our mouth, which are the parotid gland, submaxillary gland, and sublingual gland. Parotid, submaxillary, and sublingual. Sublingual must be below the tongue. Parotid gland, submaxillary, and sublingual gland. The secretions of these glands are called saliva. Then composition of saliva, daily secretion 1500 ml per day. That's a large amount. Don't you think? Yes. So pH less than 7 during resting time and approach 8 during active secretion before eating food or when we salivate just by looking into some food item. Then lingual lipase secreted by glands on the tongue. Salivary alpha amylase or ptyalin by salivary glands. Then mucins, glycoprotein that lubricate the food, bind bacteria and protect oral mucosa. Mucin, salivary alpha amylase and lingual lipase. Immunoglobulin and lysozyme attack the bacteria, which are the major constituents. Salivary amylase, lingual lipase, mucins, immunoglobulin and lysozyme. Along with that, lactoferrin bind iron and it's bacteriostatic and is bacteriostatic. So when we try to learn about the ionic composition, generally the saliva secreted by the acini is isotonic with plasma. The excretory ducts and the intercalated duct drain into them modify the composition of the saliva by extracting Na plus, Cl minus and adding K plus and HCO3 minus. So low rate of flowing saliva. So low rate of flowing saliva in saliva reaches the mouth is hypertonic by depletion of Na plus, Cl minus and rich in K plus. And the flow rate is high, there is less time for change in ionic composition. So don't forget the components, which are the lingual lipase will be there, salivary alpha amylase or ptyalin, along with mucins, then immunoglobulin and lysozyme. Some amount of lactoferrin also. Control of salivary secretion. Next topic. Stimulation of the parasympathetic nerve causes release of proteolytic enzyme kallikrein from the gland cell, which act on plasma alpha 2 globulin to form bradykinin, also cause release of VIF. This cause vasodilation of blood vessel in the salivary gland and also stimulate profuse secretion of watery saliva with low content of organic material and proteins. So stimulation of the sympathetic nerve causes secretion of small amount of saliva rich in organic constituents and mucus from submandibular and sublingual salivary glands. Earlier we learned about which are the major salivary glands situated: sublingual, submandibular and submaxilla. Don't forget that. And functions of the saliva: salivary alpha amylase, digestion of cooked starch or starch to maltose. Mucin lubricates the food, thus helping mastication and facilitates swallowing. Saliva keeps the mouth moist and serves as a solvent for the stimulation of taste buds. It minimizes the buccal infection and dental caries. Swallowing from the mouth. The food reaches the stomach by the process of swallowing as the reflex response triggered by afferent impulses in trigeminal, glossopharyngeal and vagus nerve and centers in medulla. So 3 stages: oral stage after mastication of food mixed with saliva and forming a bolus. Then contraction of the front part of the tongue presses the bolus against the hard palate, while moving in the middle pushes into the back. Swallowing begins by closure of mouth and contraction of mylohyoid muscle. So pharyngeal stage involuntarily closed coordinate the movements in the pharynx that pushes the bolus into esophagus. Esophagus stage, local stimulation in esophagus causes peristalsis. Relaxation of lower esophageal sphincter causes movement of food into the stomach. Next, food reached the stomach. So after that, food is stored in stomach, mixed with acid, mucus and pepsin, released at controlled steady rate into the duodenum. So now, food is stored in stomach, mixed with acid, mucus and pepsin, released at controlled steady rate into the duodenum. Gastric mucosa contains 3 types of glands: main gastric glands, cardiac glands, pyloric glands. Main gastric glands alveoli contains chief cells which secrete pepsinogen, parietal cells which secrete HCl and intrinsic factor. Cardiac gland secretes soluble mucus. Pyloric gland secrete soluble mucus and contains G cells which secrete a hormone of gastrin. So gastric secretion, daily secretion 2500 ml per day. Remember the amount of salivary saliva secretion in our mouth on a day-to-day basis, which was 1500 ml per day. We just increase that with 100. That is the amount of daily secretion of gastric juice. Daily secretion 2500 ml per day. So pH 1 to 2 acidic due to HCl. So electrolytes Na plus K plus M plus H plus Mg2 plus Cl minus HCO3 minus HPO4 2 SO4 2 minus. Pepsinogen. Pepsin helps to digest proteins. Rennin digest milk. Gelatinase liquefies gelatin. Gastric lipase fat splitting enzyme. Then lysozymes bactericidal enzymes. Along with that urease hydrolyze urea to ammonia. Then carbonic anhydrase etc. are present. Once again I will be reading the left side. Pepsinogen, rennin, gelatinase, gastric lipase, lysozymes, urease, and carbonic anhydrase. Along with that, mucus and intrinsic fat, which helps to absorb vitamin B12. Mucus, soluble and insoluble type. Then intrinsic fat helps in the absorption of vitamin B12. So mechanism of HCl secretion in detail we will be learning. So gastric juice is isotonic with plasma. Therefore, mechanism in the parietal cell can actively transport H plus against concentration gradient by H plus K plus ATPase in the apical membrane. Parietal cells are intracellular canaliculi and many inactive vesicles which contain H plus K plus ATPase. And these cells are stimulated, the vesicles fuse with cell membrane and H plus secreted into the lumen of canaliculi in excess of K plus by H plus K plus ATPase. Cl minus also secreted down in this electrochemical gradient either by pump or through a channel that are activated by CAMP in the apical membrane. The flow of Cl minus creates a negative potential inside the canaliculi, causing K plus to flow passively into the canaliculi. The H plus that is secreted into the lumen comes from HCO3 minus, which in turn is formed by the hydration of CO2 in the presence of enzyme carbonic anhydrase and parietal cells rich in these enzymes. The HCO3 minus formed by dissociation of HCO3 minus is discharged into the intestinal fluid by an air port in the basolateral membrane of the parietal cell that exchange HCO3 minus mainly for Cl minus. So regulation of gastric juice secretion, regulation brought by two mechanisms, nerves mechanism and humoral mechanisms. Nervous mechanisms in the sense, this mechanism include local autonomic reflexes involving cholinergic neurons and impulses from the CNS by way of vagus nerve. Vagal stimulation increased gastric juice secretion by release of gastrin releasing peptide, which increases gastrin secretion. This in turn increases acid secretion. So, released acetylcholine acts directly on the glands in the body and fundus of the stomach to increase acid and pepsinogen secretion. Later, humoral mechanism mediated by release of hormone gastrin. Psychic stimuli and digestive products of food in the stomach increase gastric juice secretion. Vagal action causes the secretion of gastric juice by parietal cells and release gastrin from gastrin cells of the pyloric antrum. And regulation of gastric motility and emptying. Gastric motility, when we learn about that, stomach has basic electric rhythm, a wave of depolarization of stomach muscle cell proceeding from the circular muscles of the fundus of stomach to the pylorus. Peristaltic contractions in the stomach is coordinated by basic electrical activity occurring in every 20 seconds. The spread of electrical activity occurs to the gate junctions. So, gastric emptying, humoral mechanism mediated by release of hormone gastrin, psychic stimuli and digestive products of food in the stomach increase gastric juice secretion. Vagal action causes secretion of gastric juice by parietal cells and release gastrin from gastrin cells of the pyloric antrum. Next, regulation of gastric motility and emptying. This is a very important topic, so please pay attention. Stomach has basic electrical rhythm, a wave of depolarization of smooth muscle cells processing from the circular muscles of the fundus of stomach to the pylorus. Peristaltic contraction of the stomach is coordinated by basic electrical activity occurring in every 20 seconds. The spread of electrical activity occurs through gate junctions. Gastric emptying, food enter the stomach usually as a mixture of liquid and solid. The kind leaving the stomach is essentially a liquid. So gastric emptying begins as soon as the large part of gastric contents become fluid enough to pass through the pylorus. Gastric emptying. So gastric emptying, food enter the stomach, which just we learned. So gastric emptying results from progressive wave of contraction which subsequently involve antrum, pylorus and proximal duodenum. Gastric emptying begins as soon as large part of gastric contents become fluid enough to pass through the pylorus. Food enter the stomach as a mixture of liquid and solid as a mixture, but leaves as a liquid. Functions of stomach. Storage function, serve as a temporary reservoir for the ingested food. Mechanical function, peristaltic movements of stomach mix the bolus with gastric juice and convert it into chyme. Digestion function, gastric juice contains many enzymes that acts on proteins, carbohydrates and lipids. Then productive function in the sense, the hydrochloric acid present in gastric juice destroys many types of bacteria entering through the food. Then hemopoietic function, intrinsic factor present in the gastric fluid juice, which we learned, which helps in the absorption of vitamin B12 earlier. So that is essential for absorption of vitamin B12. Oh, that's it. Which is an important maturation factor during erythropoiesis blood cell formation. Hematopoietic function, intrinsic factor present in gastric juice, is essential for absorption of vitamin B12, which is an important maturation factor during erythropoiesis. And excretory function, many substances like toxins, alkaloids, and metals are excreted through gastric juice. Which are the major functions? Once again, let's have a quick recap. Storage function, mechanical function, digestive function, protective function, hematopoietic function. Just remember the intrinsic factor along with vitamin B12 and erythropoiesis. Then excretory function. Let's learn about pancreas. Has got exocrine and endocrine portions. We know the exocrine portion of pancreas secrete pancreatic juice. Exocrine portion of pancreas secrete pancreatic juice. Composition of pancreatic juice, alkaline and rich in HCO3. Daily secretion 1500 ml. Similar to saliva. So pancreatic juice contains organic and inorganic substance. Organic substances are enzymes and others. The powerful protein splitting enzymes of the pancreatic juice are secreted as inactive Rose enzymes. Proteolytic enzymes are there. Trypsinogen, trypsin, by brush border enzyme enteropeptidase, which when peristalsis juice enters the duodenum, trypsin converts chymotrypsinogen to chymotrypsin and proelastase to elastase, then procarboxypeptidase to carboxypeptidase. Lipolytic enzymes are also there. Pancreatic lipase digest both cooked and uncooked starch to maltose. Pancreatic esterase converts cholesterol esters to cholesterol. Then phospholipase A2 splits fatty acids of lecithin, forming lysolecithin. Just learn all these topics just by paying some attention. Then regulation of secretion. Secretion primarily controlled by hormones. Secretion acts on the pancreatic ducts to cause copious secretion of alkaline pancreatic juice rich in HCO3 and poor in enzyme. CCK acts on Athena cells to produce enzyme-rich pancreatic juice, but in low volume. Cold cytokinin CCK. The next functions of pancreatic juice has digestive and neutralizing function. Trypsin convert proteins into proteoses and polypeptides. Chymotrypsin hydrolyzes the proteins into polypeptides. Then carboxypeptidase convert polypeptides to amino acids. Nucleases digestion of nucleic acids. Then elastase digest elastic fibers. And collagenase digestion of collagen fibers. Next, digestion of lipids. Just once again have a recap about the digestion of proteins. Trypsin, chymotrypsin, carboxypeptidase, along with that nuclease, elastase and collagenase. Next, digestion of lipids. Pancreatic lipase converts triglycerides to monoglycerides and fatty acids. Cholesterol ester hydrolase converts cholesterol ester to free cholesterol and fatty acids. Then digestion of carbohydrates. Pancreatic amylase converts both cooked and uncooked starch to maltose. Then regulation of secretion, primarily controlled by hormones. Secretin acts on the pancreatic ducts to cause copious secretion of alkaline pancreatic juice rich in bicarbonate and poor in enzymes. CCK acts on the acina cells to cause the production of pancreatic juice rich in enzymes with low volume. Then liver and gallbladder. Being the largest gland in the body, the major functions of the liver is a filter between the blood forming from the GIT and blood in the rest of the body. Blood from the intestine and other viscera reach the liver through portal vein. So, blood percolates in the sinusoids between the plates of hepatic cells and drains it to hepatic veins which enter the inferior vena cava. Then functions of liver, synthetic functions. It synthesizes most of the plasma protein, especially albumin, some clotting factors, fibrinogen, prothrombin, factor 5, 7, 10, etc. Enzymes like alkaline phosphatases, SGOT, SGPT, SICD, I don't know what that is. So, urea and cholesterol, these are some alkaline phosphatases. Next, metabolic functions. On carbohydrate metabolism helps in synthesis, storage and release of glucose by the following processes: glucogenesis, glucogenolysis, gluconeogenesis. On protein metabolism, synthesizes plasma proteins, blood clotting factors, enzymes, urea and lipoproteins. In fat metabolism, beta oxidation of fatty acids, esterification of fatty acids to form triglycerides, synthesis of lipoproteins, cholesterol and phospholipids. Next, bile secretion, synthesis of bile salts and bile acids from cholesterol, conjugation of free bilirubin, bile pigments with uridine diphosphate glucuronic acid. Detoxicating and protective function by complete destruction of drugs like nicotine and short-acting barbiturates. By the activity of Kupffer cells helps in immune mechanism. By conjugation of unwanted substances can be excreted in the urine. Storage organ for glycogen, fat, protein, vitamin A and B12. Hormone inactivation like cortisol, aldosterone, insulin, then glucagon, testosterone and thyroxine, and then antibacterial action. The bile is secreted continuously by hepatic cells. Daily secretion 500 to 1000 ml. pH 7.8 to 8.6, water 96%. Composition of bile salts, sodium and potassium salts of bile acids, bile pigments, biliverdin and bilirubin, fatty acids and cholesterols, alkaline phosphatase enzyme. Functions of bile salts: reduce the surface tension and help in emulsification of fat for digestion and absorption. They form the micelles with lipids which can be easily absorbed, act as a laxative by stimulating peristaltic movements, prevent gallstone formation by keeping the cholesterol and lecithin in solution. Then gallbladder, bile flows into gallbladder when sphincter of OD closed. In the gallbladder, bile gets concentrated by the absorption of water and stored for release when needed. Then regulation of biliary secretion. When food enters into the stomach, the resistance of sphincter of OD reduces. Fatty acids and amino acids in the duodenum releases CCK or called cytokinin, which causes gallbladder contraction. Bile production increased by stimulation of vagus and by secretion hormone. Then small intestine, it is the major site for digestion and absorption of carbohydrates, proteins and fat. The intestinal contents mixed with intestinal juice, pancreatic juice and bile, mucosal surfaces adapted to provide huge area for absorption by villi and microvilli. Next, composition of intestinal juices. Daily secretion 3 liters, excessive amount of intestinal juice, just 3 liters. pH 7.6, water about 98% to 6%, 98.6%. Enzymes enterokinase, repsin, intestinal lipase, cholesterol esterase, etc. Then intestinal motility. Other than MMC and BER, there are 3 types of smooth muscle contraction. Peristalsis propels the chyme towards large intestine. Segmentation contractions move chyme to and fro and increase its exposure to mucosal surface. Then tonic contractions, which are the major contractions: segmental contraction, peristalsis, and tonic contraction. Prolonged contraction, which isolate one segment of the intestine from other. Tonic and segmental. Okay. So intestinal digestion in small intestine, carbohydrate digestion. Sucrase converts sucrose to glucose and fructose. Maltase convert maltose to 2 molecules of glucose. Lactase convert lactose to glucose and galactose, I guess. So fat digestion, intestinal lipase converts fats into monoglycerides. Next, protein digestion, peptones and polypeptides are converted into amino acids by pepsin. Nucleic acids are converted by nuclease into nucleotides. Large intestine, the diameter is more than that of the small intestine. Mucosal surface of colon is smooth and no villi on mucosa. Main function of the colon is absorption of water, Na+ and other minerals. By removing 90% of the fluid, it converts 1000 to 2000 ml of acetonic chyme that enters the large intestine to about 200 to 250 ml of semi-solid feces. 200 to 250 ml of semi-solid feces. Large difference. Let me just see 1000. So movements of large intestine. It includes segmentation, contractions, and peristalsis. A third type contraction that occur only in colon is mass action contraction. Mass action contraction, in which there is a simultaneous contraction of smooth muscles over large fluorescent areas. Then absorption in the colon. Absorptive capacity of mucosa very high. Na+ is actively transported out of the colon and water follow along the osmotic gradient. Normally there is innate secretion of K+ and HCO3- in the colon. Feces, stool remains in organic materials, undigested plant fiber, bacteria and water. Their composition is relatively unaffected by variation in diet because large fraction of fecal mass is of non-dietary origin. Then intestinal bacteria. Bacteria in the GI tract can be divided into 3 types. Some of the pathogens that cause disease. Others are symbiotes that benefit the host and vice versa. Mostly commensals, which have no effect in host. Symbiotes, pathogens, and commensals. Commensals I'm showing. There's no effect at all in the host. Then at birth, the colon is sterile. The brown color of stool due to pigments formed from bile pigment by intestinal bacteria. It plays a role in cholesterol metabolism. Poorly absorbed neomycin modified flora, lower LDL and cholesterol level. Now let's have a conclusion about all this topic we just learned. So let's have a quick recap. Digestive system is the portal through which nutrients, minerals, vitamins and fluids enter the body. The ingested food is digested by the coordinated activity of various organs in the GI tract with their enzymes and hormones. The products of digestion are absorbed from the intestine and waste materials are expelled out. Proper functioning of digestive system is essential for normal growth and development. Then summary of digestive system. Living organisms require nutrients and oxygen for survival and growth. We get all these things from the food, but the body cannot utilize the food material in the form as we eat. It has to be broken down into small particles by the process of digestion. Only then it can be absorbed. Parts of digestive system. Digestive tract is continuous, starting from the mouth, passing through the pharynx, esophagus, stomach, small intestine, large intestine, rectum, and finally into anus. Accessory organs of digestion: the teeth, tongue, salivary glands, liver and gallbladder and pancreas. General organization of the digestive system. In general, the wall of the digestive system from posterior pharynx to the anus has following layers from inward to outward: mucosa, submucosa, muscularis and serosa. Mucosa, submucosa, muscularis and serosa. We learned this. Then intrinsic innervations. The 2 major network of nerve fibers intrinsic to the GI tract are the mesenteric plexus and the Meissner's plexus between the middle circular layer and the mucosa. Functions of the GI tract is responsible for breaking down the food particles and supplying the body with water, nutrients and electrolytes needed to sustain life, which is brought by the following processes of ingestion, including placing food into the mouth, chewing the food and breaking into smaller pieces of mastication, moistening the food with salivary secretion. And digestion, during which the food particle is broken down into smaller particles by the grinding action of the teeth and degraded by digestive enzymes into absorbable nutrients. Then absorption, during this process, the nutrients, water and electrolytes are transported from the GI tract into the circulation. Digestion. During this process, the undigested food along with then various secretions and loaded off epithelial cells from the GI tract passes into the rectum and form the feces which are voided from the anus at periodic intervals. Next, process of digestion and absorption. Mouth and salivary glands. In the mouth, food is mixed with saliva, and by the process of mastication, large food particles are broken down into smaller ones. The food particles form a bolus and is propelled into esophagus. Next, functions of the saliva. Salivary alpha amylase or ptyalin, digestion of cooked starch to maltose. Mucin lubricates the food, thus help in mastication and facilitates swallowing. Saliva keeps the mouth moist and serves as a solvent for the stimulation of taste buds. It minimizes the buccal infection and dental caries. Swallowing from the mouth, the food reaches the stomach by the process of swallowing. It is a reflex response triggered by affected impulses in trigeminal, glossopharyngeal and vagus nerves and centers in medulla. Next, stomach. Food is stored in stomach mixed with acid, mucus and pepsin, released at a controlled steady rate into the duodenum. In gastric glands, alveoli contains chief cells which secrete pepsinogen, parietal cells which secrete HCl and intrinsic factor. Gastric glands secrete soluble mucus. Pyloric gland secretes soluble mucus and contains G cells which secrete a hormone gastrin. So functions of stomach include storage function, protective function, mechanical function, hematopoietic function. Just remember the intrinsic factor along with vitamin B12, and digestive function, excretory function. Next, pancreas has got endocrine and exocrine portions. The exocrine portion of pancreas secrete pancreatic juice. Then functions of pancreatic juice has digestive and utilizing function. Digestion of protein, trypsin converts proteins to proteases and polypeptides. Chymotrypsin hydrolyzes the proteins to polypeptides. Carboxypeptidase convert polypeptides to amino acids. Nucleases, digestion of nucleic acids. Elastases, digest elastic fibers. Collagenase, digestion of collagen fiber. Then digestion of lipids. Pancreatic lipase converts triglycerides to monoglycerides and fatty acids. Cholesterol ester hydrolase controls cholesterol esters to free cholesterol and fatty acids. Then digestion of carbohydrates. Pancreatic amylase converts both cooked and uncooked starch to maltose. Then liver and gallbladder. Being the largest gland in the body, the major functions of the liver is as a filter between the blood coming from GI tract and blood in the rest of the body. Then functions of the liver include formation and secretion of bile, nutrient and vitamin metabolism, inactivation of various substances like toxins, steroids and other hormones, synthesis of plasma proteins, immune response by Kupffer cells. The bile is secreted continuously by hepatic cells. Next, functions of bile salts reduce the surface tension and help in emulsification of fat by the digestion and absorption. They form the micelles with lipids which can be easily absorbed, act as laxative by stimulating peristaltic movements, prevent gallstone formation by keeping the cholesterol and lecithin in solution. Then gallbladder. Bile flows to gallbladder when sphincter of ovary closed. In the gallbladder, bile gets concentrated by absorption of water and stored for release when needed. In small intestine, it is the major site for digestion and absorption of carbohydrates, proteins and fat. The intestinal contents mixed with intestinal juice, pancreatic juice and bile. Mucosal surface is adapted to provide large area for absorption by the villi and supervilli. Then digestion in small intestine. Carbohydrate digestion. Sucrase converts sucrose to glucose and fructose. Maltase convert maltose to two molecules of glucose. Lactase convert lactose to glucose and galactose. Fat digestion. Intestinal lipase convert fats into monoglycerides. Then protein digestion. Peptones and polypeptides are converted into amino acids by ribosome. Nucleic acids are converted into nuclease by nucleotides. Then large intestine. Mucosal surface of colon is smooth and no villi on mucosa. Main function of colon is the absorption of water, Na+ and other minerals. And absorption in the colon. Absorptive capacity of mucosa very high. Na+ is actively transported out of the colon and water flows along the osmotic gradient. Normally, there is net secretion of K+ and HCO3- in the colon. Intestinal bacteria. Bacteria in the GI tract can be divided into three types, which we just learned: pathogens, symbionts, and commensals. Okay. There are pathogens that cause disease, others are symbionts which benefit the host just by producing some vitamins, etc., and also helping in digesting cellulose such kind of processes. Then vice versa, most commensals which have no effect in host. At birth, the colon is sterile, the brown color of stool due to pigments formed from bile pigments by intestinal bacteria. It plays a role in cholesterol metabolism, poorly absorbed neomycin modified flora, lower LDL and cholesterol level.